My name's David Neal. For the previous 12 years, I worked in Cambridge as Professor of Surgical Oncology and a group leader in the Cambridge Institute, largely studying prostate cancer. Recently, I've been working with Ros Eels in the audience here and Andy Lynch, doing ICGC studies on prostate cancer. And for the past year, I've moved to work with Elsevier as their Senior Vice President in global research, really developing new tools. In fact, doing some work on big data around recommendations and the use of Scopus, and actually producing new tools for researchers as well.
Thank you. And Francesca?
Hi, I'm Francesca Buffa, and I head the computational biology and integrative genomics group in Oxford. I come from physics originally, and then I slowly went to biostatistics and then bioinformatics. And really our work focuses very much on discovering signatures of aggressive tumor that respond poorly to various treatment, and translating these and try to understand how to translate them to the clinical setting. And so, for example, how many things do we need to measure in the clinical setting? And we started from gene expression historically. We are now adding all the various components that are available, and even more the question is really pressing on what do we need in the clinical setting. We don't want to miss anything that is needed. On the other hand, how is it feasible to measure everything in the clinical setting, and can we cope with that complexity, on how do we reduce it?
So I had a few thoughts when I was listening to Wolfgang's talk. I mean, I think clearly there's a lot of public debate and interest about reproducibility. And I think quite often there's a bit of naivety in this, in that if you look, so certainly at Elsevier, for instance, we are now producing executable papers along the lines that you've suggested, which works fine if the data are correct and the methods are accurately published. But for you in the audience, many of you will be working on really cutting edge technologies and developing new approaches to sequencing, where invariably there are going to be day-to-day variations in measurements and the ability to record things. So I think we really need to distinguish between problems with the method, where they're not clearly published and you can't repeat the experiment, with, at the other extreme, technically very, very difficult experiments at the cutting edge. So in one way, you know, although there is a lot of concern about this, time does tell. I mean, in terms of proving the quality of research. So you could take a fairly, not quite relaxed view on it, but there will be certain experiments that will only be shown when they stand the test of time. So I think it's a very complex area, and you know, running from, you know, one extreme real issues about research integrity and quality control, to right at the other, you know, day-to-day variations and experiments. And I think they're often lumped together in a very crude way.
Francesca, would you like to add something?
Yeah, I think it links pretty much with what is said. But one aspect, for example, is that our biological understanding is changing. Yeah, and so, for example, if we want to use signatures that were developed 10 years ago, and we try to, a very simple thing, we try to re-annotate them. This is an exercise we've just done, actually, for hypoxia signatures. And you try to re-annotate them, and you find out that the content is actually different, not because anything was wrong, but because the understanding of what is a gene and which are the genes and how do they work has changed. Yeah? And so there is part of reproducibility, but it's not the standard part of reproducibility we are all used to discuss. Nothing went wrong. It's just as biology evolves so quickly, we need to be able to really find ways of addressing this continuous evolution in the work and implementing it down to the clinic as well.
Well, I mean, I'd like to echo something. I actually think that typically the biggest problem that I've seen often comes with the data. So even when the method is very good, the method is making a lot of assumptions about how well the data is behaving. And this is very complex data that often doesn't behave well. So actually the mismatch between perhaps completely reproducible and robust method to an experiment that went awry. So there needs to be more QC between method and input data to really keep the integrity of the science, and it's more often the data rather than the method that's at fault.
Wolfgang, do you want to add
Yeah.
any?
Well, I mean, I think in some sense, I think it's simpler than what David worried about. If the task is basically to take whatever is the raw data for this experiment that came out of the machines, and then basically converting it into what people then put as the figures and tables of their paper. I think that's a well-defined task, and that can actually be addressed by a reproducible document. Of course, there are challenges, especially if the computation is needed. Especially in genomics, they can take sometimes weeks and months on a computer, or you might need specialized equipment. And then, of course, mirroring that in a reproducible document may be very difficult.
So I'm Ed Curry, I'm from the Division of Cancer at Imperial College. Do you think it sets a bad precedent allowing journals that publish, very high-impact journals which publish a lot of genomics papers, to put all of the methodology that underpins all of the findings in that paper, leave them to the supplementary to say, actually, they're not that important for this paper, and that most people who will read those probably will not go through the supplementary information, so can't possibly understand, really, the nuances of the findings that are presented? And do you know, is there anything that we can do to stop that?
Go on, Pat.
I actually think that's a huge problem. I actually find the big journals often have tabloid advertisements where I simply flip the supplement to begin with. Now, the fact that it's pushed to the supplement in itself might be, arguably bad or good. I think the really bad part is that this supplement is less vetted, is less edited, is less clear, is often missing critical information. So even when you go to the supplement, it's terse, and you can't even reading the supplement, I can't figure out what the authors actually really did. And that's when things really go bad.
So do you think there's anything we can do to stop that as a community?
I think maybe it would be good to deconflate two things that are currently quite conflated. I think the big journals have this argument that they have a finite number of pages which they can have every week, and so the space is limited. And I think that argument doesn't cut it anymore these days with the web and the internet. Of course, the real argument, the other argument is that people have a finite amount of attention, and that most readers do want some kind of high-level summary of the results before they dig into the details.
Yeah, I think I'd echo that. I mean, in a sense, a lot of what we're debating is how journals have evolved, how scientific communication has evolved, and the very high impact journals that we've been talking about that Cell you had many of your papers this morning were from Cell. Quite often, as you say, people want a high level summary to read through them if they are going to read through a paper journal. But increasingly, I think that model is changing. People are searching for particular articles on the internet that necessarily scan through the whole journal. But the fact is, I think unless somebody's going to be very interested in a particular paper, they're not going to go through the supplementary material in detail. So I think the answer is a bit like you were saying, actually, that maybe journals should be much more concerned in making sure that the data are uploaded at the time of publication, that those are reproducible. But speaking from my Elsevier hat, I know that PLOS, when they insisted recently that everybody uploaded their data at the time of publication, they saw a pretty large drop-off in submission rates. So that there's this tension between a general view in the community that data should be made more open, which, by the way, to be clear, Elsevier is very supportive. I'd like to speak on their behalf. That tension there, and individual researchers who are not always as keen.
Do you have any comments on views?
I'm not really sure what to. It's something that, as a bioinformatician embedded within a biology institute, I find it very difficult presenting papers or sitting in talks where people are mentioning some paper that they've looked at, they want help interpreting, because all of the methodological detail that underpins all of the results that are being presented are left embedded, hidden in supplementary information. And as Dana said, you do often have found instances where the main document says refer to supplementary methods, which say as carried out previously, refer to another paper, where you go to that paper, go to its supplementary methods, and then it's pointing to a different paper where they haven't used any method that's related to the first one. And they've just effectively led a trail of obfuscation in order to say, actually, we're just waving our hands as to what we really did here, and it's impossible to reproduce it, and it's impossible to work out what they're actually saying.
So what else have you for resolution?
Well, I think we are doing very much the sort of things that we were talking about earlier, and we shortly will be, I should say, in Cell Press. There will be a pilot out later this year where people will be able to upload all their data at the time of submission. So it's an area we're very concerned about.
Francesca,
Yeah, just maybe also another way of, because one of the problems with these, I mean, when you go to big papers, one of the problems is that they're usually led by a question, and then the methodology is left a little bit behind, and also maybe in the team, the people providing the methodology are not really pushed because they know it's going to go in the supplementary methods, right? So they're not really pushed. Maybe there should be a bigger push on the methodology, and so maybe more kind of matched publications, like if you have a new study with new methodology, you sort of encourage, I mean, the editors should pick this up and encourage more kind of collaborative publications of paper. I mean, ENCODE is already a good example of that, but maybe there should be more of that, and even on the methodological side.
and then we'll
I'd like to stress that it's not an issue of making the data public, but as Kieran said, it's an issue of making the methods clear, well-written, and present. So the problem is actually how do you get from that data, which you can make public, to the stated results in the paper. When you go to the supplement, it's often missing, poorly written, and there's just no way you can try and figure out how the author went from the data, even if it's public, to the conclusions that they reached, because there's, it's really swept under the rug. And I don't mind if it's in the supplement. I just want there to be very high quality standards for that supplement.
So one practical implication from this discussion that I think will then have to follow is also a revision of the peer review system, because the current way we review papers, I think it's just not able to bear the workload of going through 100 pages of supplement and rewrite the code and everything. And we will probably need some combination of what we have now and post-publication review on these internet-based sites.
Great, thank you. Let's move on to another question. Could you introduce yourself?
Jonathan Pearson, MRC. It's actually a related question. I mean, is there a role here for reporting guidelines, which I know have been developed in other fields to try and advance, what are the minimum reporting elements, methodological needs that should be reported in papers? Which is something that the community, and by the community I would certainly include the journals, the funders, this isn't just the academics, it's the whole issue of reproducibility needs us to work together.
I think that is a very good point. I mean, we had remark, we had all the various, why not a methodological sort of checklist. At least it would guarantee some minimum, yeah? And then maybe it could even just, another thing that could be done maybe is a maybe cross-journal an infrastructure where you can run and upload your code, because another problem from the publishing point of view is that, okay, you put your code there, but then there is a new version of bioconductor, everything fails, and so maybe a more structural way of doing it between journals, so a portal where people could run the code, why not? So this kind of technological help here in the review process. And I'm suggesting.
Well, speaking of code, and this might be less relevant for this audience. I don't know what the situation is in the UK, but you develop code. It's developed often by a graduate student rather than a professional programmer. It's developed at a certain point in time, and I know that at least in the US, it's almost impossible to get money to maintain code, which is actually a pretty important component.
Any comments or questions? No. Maybe we can move on to this aspect of the dimensionality. You touched on, both the talks touched on the availability of many, many dimensions or variables. but at small sample sizes, and that poses a challenge. Maybe I can ask you to extend on what you
Sure. So first of all, there's dimensionality. So any one of these technologies, which we discussed, we have 3 billion base pairs, 20,000 genes, about a couple 100,000 regions that are known regulatory elements. So the genome itself has hundreds and thousands to even millions of dimensions in terms of what nucleotides we need to look at, gene expression. It's a completely different type of data. We have, again, tens of thousands of genes, hundreds and thousands of transcripts because these genes differ in their splicing. So each of these data types that I mentioned has between tens of thousands to hundreds and thousands of dimensions, and the heterogeneity that both of us mentioned. You have different data types. You have to look at the DNA, the gene expression, the epigenome, methylation status, histone modifications. These are very different molecular features. So to take all these different types of features that are molecularly different, each of them existing in tens to hundreds of thousands, and to try and understand this very, very complex domain with typically hundreds to low thousands of samples is the big challenge of genomics.
Wolfgang.
Actually, I think it's where all the fun lies in this field is actually dealing with those many dimensions.
Oh yeah, that's why I'm there.
That's
But that's the hard
exactly.
part.
Yeah.
That's the crux of it.
Yeah, because basically what you need to do is to use sort of what's called Bayesian models or regularization over the last 15 years, the field has developed a lot of approaches here, and I think we pretty well know how to deal with this now technically through Bayesian modeling or regularization. And this is actually now where you can sort of input other information or prior knowledge or ideas about the structure of the system to then basically overcome this this cause of high dimensionality. So that's...
Yeah, David?
Yeah, I just a couple of questions, really. I mean, one of the things that struck me as a clinician listening to this is that often we're spending a lot of time getting really accurate genomic, proteomic data, but the clinical data are really very, very fuzzy. They're difficult to disambiguate. They're difficult to pull out from the electronic record, and I think that's going to be a real challenge, I think, for realizing some of the ambitions that we've heard about. I had a question for Dana, really. I mean, is it perhaps no accident that both of you were working on fluid malignancies in a way? And I was thinking that work that we've done on prostate cancer, where maybe in the primary cancer, there may be only one in a million cells that matter. And how are we going to get over this problem of sampling the right cell? The other thing that struck me as a surgeon as well, we used to be very naive that if you had a localized tumor, you would cut it out. I mean, that's clearly totally incorrect. And one of the things that really interested me about you pointing out tumor infiltrating cells in the primary cancer is what sort of biological interaction is happening when you actually remove a primary tumor. Why does surgery work, actually, in some instances in cancers that we know will be shedding cells throughout their whole life? And the final point I had for Dana, what about the protein aspect, post-translational modifications that we've concentrated on genomics? How important are those?
All right, so I have a long list. So I think one of the problems with clinical data is, again, the noise in the data. So we have very ordered, structured, controlled data sets for some of the more in vitro systems. But I've worked a lot with clinical data in the past 5 years, and how the tumor and the sample is extracted from the patient, how long it stays on a slab before someone addresses it, how that is treated in each, because when there's a patient on the table, then that patient's life rather than the integrity of my sample is what matters. On the other hand, it would be nice, and I've seen the difference in the data, when there's a person dedicated there actually to handle that piece of tumor and to handle the data, and not the patient. So processing the sample, dissociation. So I've also been working with primary samples, and yeah, the blood tumors, they're really nice because they come already in suspension, and they're not that disrupted or upset when we're taking them out, even though they don't like being out of the body as well. But when you're taking a physical tumor and breaking it apart, RNA, which I find the most valuable piece of information, gets rapidly disrupted and changes. As you pull this tumor apart, we try and do everything on ice, and really, how do you get these cells from clinical samples out of the patient, separated and measured with minimal disruption, is one, if not the biggest challenge of translating everything we talked about here to clinic. That's number one. In terms of how do we get to the number of cells that you wanted, so again, let's say we solved getting the patient out and getting it disrupted, and there's a lot of people working very, very hard on that. My approach to it is taking the single-cell RNA-seq and the indrop, which I just described, which we have working in our lab, to look exploratory, what are the features and the markers and the genes that matter, and here I can look genome-wide. Then I can say, okay, here are about 50 features that I think are the important ones, and I might be wrong, but maybe we can do that. Then there are other technologies such as CyTOF, which really can give you 5-10 million cells in a single experiment. And once you've narrowed it down to those 50 markers you need to be looking at, you can switch to a technology which can give you millions of cells, and then you can get to those real rare cells. And I think that should be the pipeline: exploratory, smaller signature, another technology to go deeper and deeper to get that rare cell. And yes, I mean, CyTOF actually does work on antibodies. It's one of their limitations. It does look at proteomics and even phosphoproteomics, and I do see a lot of interesting things once I look at p-ERK status. But RNA is actually very powerful because almost any perturbation in the cell, because the circuit is so interconnected and everything is connected to connected, if you alter its metabolism, if you alter its external phenotype, if you alter the force and the pressure that's put on the cell, almost anything you do to the cell is going to have an impact and a signature in the RNA if you know how to interpret it as such, which is also one of the reasons why this association is so hard, because that process immediately impacts the RNA. And that was a lot, but there were many questions.
Yeah, thank you.
One final question, comment here.
Well, I just wanted to ask you, what N of patient number would you regard then as being appropriate, given what you just said?
So I'll repeat the question. What N of patient number do you feel is appropriate given what you thought?
So if I look at it as a computer scientist and as a statistician, then we're hopeless. Like, N needs to be millions. One of the things that we luck out on is often they're very big signals in biology, so things that, according to statistical theory, shouldn't work. When the signal is strong enough, you could pick it up with smaller N. And I have picked up interesting signals where N is in the dozens, but really you need big N, and I think that is another big thing. In order to really make clinical genomics or clinical personalized medicine, we're actually going to have to get the clinical N of the worldwide network. So everyone's going to have to share data across samples, and there's no single initiative, single hospital, single country that will be able to contribute enough. And if we standardize some of these things, so we could do meta studies across cohorts, hospitals, and countries, that's when personalized precision medicine will become a reality.
Great.
I just
Final comment
so
to wrap up.
is absolutely bang on what you're saying, because we are trying to experimental phase we can go up with numbers. Yeah, in clinical trials we can't. So we have to be on one side much more careful to go and plan the correct clinical trials. On the other side, maybe we have to start to cooperate much more across trials. to really have the number of patients that we need. So trials have to become a little bit less hypothesis and investigator-led. They have to become a little bit more shared trial in the community and evolving trial as well. I mean, there are methods that are being developed to deal with these, Bayesian kind methods, but also the mentality of, it's not just the methods that need to change, it's also the mentality of organizing trials that maybe needs to change, and we need to share patients across trials as well. Or having a fast update of our clinical knowledge and the way we record clinics. So there has to be much more communication as well between the basic science and the way we record things in the clinic, because that can increase the power enormously.